Vitamins and minerals, folate. We will now further discuss folate and its role in various reactions involved in amino acid metabolism, particularly methionine synthesis. We'll additionally discuss the RDA for folate and deficiency symptoms. Here we see the process of histidine catabolism. We previously mentioned that histidine catabolism is involved with the reaction of 1-carbon metabolism converting tetrahydrofolate to 5-formamino tetrahydrofolate. We see back on this larger diagram of 1-carbon metabolism the conversion of tetrahydrofolate to 5-formamino tetrahydrofolate. We see here the catabolism of histidine. We'll discuss in the next slide the conversion to FIGLU and also glutamate. So here reviewing in more detail this process of histidine catabolism and involvement of folate, we see that in the catabolism of histidine, it is first deaminated, removing ammonia. From there we have conversion to this urocanic acid, and then to the formamino glutamate or FIGLU. So from here, with tetrahydrofolate, FIGLU is converted to glutamate. We also need the formamino transferase enzyme, which again converts the formamino glutamate or FIGLU and tetrahydrofolate to glutamate. In the process, tetrahydrofolate is converted to the 5-formamino tetrahydrofolate. We can see here with folate deficiency or low folate status, what occurs is that this tetrahydrofolate will lower. So we have less tetrahydrofolate. So with less tetrahydrofolate, then we will not be able to adequately interact with the FIGLU to form glutamate. So with lower tetrahydrofolate and inability to convert FIGLU then to glutamate, the levels of FIGLU in the body will increase. So FIGLU is going to accumulate, and when accumulated, it is actually going to be excreted from the body in the urine in high concentrations. And we can see that actually this would be a potential method of measuring folate status, is evaluating the levels of FIGLU in the urine. Next, we will review in more detail the metabolism of serine and glycine. We can see that serine and glycine can be converted to one another. We have this key enzyme serine hydroxymethyltransferase, and the conversion of serine to glycine will involve the transfer of a methyl group. Again, this is reversible. PLP, this derivative of B6, is also going to be a cofactor assisting the activity of this enzyme. We can see in this larger diagram that the conversion of serine to glycine involves tetrahydrofolate to 5,10-methylene tetrahydrofolate. So we see those intermediates in the reaction as well. And you can also see the PLP required for this process. So we can look in more detail at the transfer of the methyl group in this process. So we see serine, we see glycine, we see tetrahydrofolate, and the 5,10-methylene tetrahydrofolate. So just take a moment and review this reaction to determine which of the two, serine versus glycine, will be containing the methyl group and then transferring it to the folate. So you can see here that serine is actually going to be initially containing the methyl group. You can see that serine, which contains the methyl group, is going to transfer the methyl group to tetrahydrofolate. After serine transfers its methyl group to tetrahydrofolate, the THF will then become 5,10-methylene tetrahydrofolate, and then serine is converted to glycine. Glycine does not contain the methyl group. So we have the transfer from serine to the tetrahydrofolate, which then becomes the 5,10-methylene tetrahydrofolate, and then the glycine does not contain the methyl group. For glycine to be converted to serine, the glycine will pick up the methyl group from the 5,10-methylene tetrahydrofolate to become serine, and then this will be demethylated to become THF. Next, we're going to discuss in more detail the synthesis of methionine. So for the methionine synthesis, we're going to see where this is going to be occurring and the intermediates involved. We see again in this diagram, we see methionine. So the detailed pathway that we will be reviewing is actually going to demonstrate the THF conversion to 5,10 methylene THF, and then the 5,10 methylene THF to 5-methyl tetrahydrofolate, and then the interaction between 5-methyl tetrahydrofolate and homocysteine to form methionine. So we're gonna see this almost triangle of reactions involved in methionine synthesis. So just a few additional notes before we review the diagram. So other intermediates involved will include this S-adenosyl methionine, also the S-adenosyl homocysteine. Also, we're going to see the involvement of cobalamin and methylcobalamin in the transfer of the methyl groups. This cobalamin is going to be the vitamin B12 involved in this process. So here's the detailed diagram which leads to the eventual conversion of methionine. We see a number of intermediates and enzymes involved. Also part of this diagram, it's broken up by first the roles of folate, and then we see the role of the cobalamin or vitamin B12. And then in this portion of the diagram, we see the conversion of homocysteine to methionine, and then the back conversion from methionine to homocysteine. So we're going to go step by step through this process. We see step 1, step 2, step 3, and these steps are going to involve primarily of transfer of a methyl group. And the goal for this process is to take the methyl group that is bound to the 5-methyl tetrahydrofolate and transfer it to homocysteine, and with that transfer, homocysteine then becomes methionine. So in the first step of the reaction, we see 5-methyl tetrahydrofolate interacting with this cobalamin, and the 5-methyl tetrahydrofolate with cobalamin will become methylcobalamin and tetrahydrofolate. And we can actually see in this diagram, so the first step of the reaction is 5-methyl tetrahydrofolate plus cobalamin will then be the tetrahydrofolate plus methylcobalamin. And what I've done is underline the intermediates that contain the methyl group. So we see here the 5-methyl tetrahydrofolate first contains the methyl group, and then that's transferred to the methylcobalamin. The next step of the process is going to involve a reaction between the methylcobalamin and homocysteine. So methylcobalamin and homocysteine will then interact. It requires the activity of methionine synthase, and from there our products will be methionine and cobalamin. So we see the reaction drawn out here once again, as well as the transfer of methyl groups. So we saw before that the methyl group is originally a component of methylcobalamin, and then it's transferred to homocysteine to then become methionine. And again, you want to keep in mind the activity of this enzyme, methionine synthase. So the next step of this reaction is going to involve the conversion of tetrahydrofolate to 5-methyl tetrahydrofolate, because for this process to continue, we need to resupply this 5-methyl tetrahydrofolate so that the process can start over again. So this third step is going to involve the conversion of tetrahydrofolate to 5,10 methylene tetrahydrofolate. For that to occur, tetrahydrofolate will need to interact with serine, which we talked about previously has a methyl group attached. So serine will transfer its methyl group to tetrahydrofolate to form 5,10 methylene tetrahydrofolate, and then when serine is demethylated, it's then converted to glycine. From there, we have activity of this key enzyme that we've discussed previously, the methylene tetrahydrofolate reductase. And with the activity of this enzyme, we have conversion of the 5,10 methylene tetrahydrofolate to 5-methyl tetrahydrofolate. We see that reaction in this diagram, so the tetrahydrofolate plus serine will then be converted to glycine, and 5,10 methylene tetrahydrofolate, and then with the activity of the methylene tetrahydrofolate reductase enzyme, we have conversion to the 5-methyl tetrahydrofolate. And we'll see the transfer of methyl groups. So first serine contained the methyl group, and then that was transferred to the tetrahydrofolate to form 5,10 methylene tetrahydrofolate. And then we have the 5-methyl tetrahydrofolate also containing the methyl group. Lastly, what is needed is the conversion back of methionine back to homocysteine. So there's a series of steps involved in this process, but the key step to keep in mind is this step here where we see the S-adenosyl methionine converted to the S-adenosyl homocysteine, because it's in this step that the S-adenosyl methionine will donate its methyl group to this methyl group acceptor, and then we have conversion to the S-adenosyl homocysteine, which no longer contains the methyl group. Here, this methyl group acceptor now contains the methyl group. So we see methionine to SAM to SAH to then homocysteine. So just a few more comments on this single nucleotide polymorphism of this gene coding for the methylene tetrahydrofolate reductase enzyme. So there's a subportion of the population that has this polymorphism, reducing the activity of this MTHFR enzyme, which then lowers the conversion of the 5,10 methylene tetrahydrofolate to 5-methyl tetrahydrofolate. So with this SMP, we have a lower conversion to 5-methyl tetrahydrofolate. So we have lower 5-methyl THF levels. And so with lower 5-methyl THF, we have reduced conversion of homocysteine to methionine, which elevates homocysteine and reduces methionine. And this may be problematic because in evaluation of cardiovascular disease, homocystinemia or elevated homocysteine levels is actually associated with negative effects to cardiovascular health. And so this is something that may be an additional outcome of this homocystinemia. This polymorphism is also associated with neural tube defects in women during pregnancy. So an individual with lower folate activity, lower activity of this enzyme, may also be at risk for neural tube defects. So we'll look at interactions between folate and vitamin B12. We saw the involvement of cobalamin in transferring the methyl group from 5-methyl tetrahydrofolate to homocysteine so that homocysteine can become methionine. And that's, of course, with the activity of methionine synthase as well. However, with low vitamin B12 status, so if someone has a deficiency in B12, the cobalamin cannot act to transfer the methyl group from 5-methyl tetrahydrofolate to homocysteine, and this is called the methylfolate trap. So that methyl group is trapped with folate, with the 5-methyl tetrahydrofolate cannot be transferred to the homocysteine, which may thus affect also methionine synthesis. We'll see now the RDA for folate. We see that in adults it's 400 micrograms per day, in pregnancy it's 600 micrograms per day, and in lactation it's 500 micrograms per day. We'll see key deficiency symptoms and risk factors. So those with low folate status will be at increased risk for developing megaloblastic macrocytic anemia. So it's a certain type of anemia leading to larger red blood cells. Those with this single nucleotide polymorphism will be at increased risk. And we see that, interestingly, in the US, there was a mandatory fortification of folic acid to grains in 1998, and as a result, there's been reduced incidence of neural tube defects.